Welcome children. In our last class we discussed about the contribution of different scientists for the structure and chemical nature of DNA, and also we discussed something about RNA, and today we will be discussing the properties of genetic material. So properties of genetic material means what criteria must be fulfilled by a molecule to become a genetic material. So already we have discussed about the various results of the scientists Griffiths and Harsich's experiment. In both the cases they have proved that DNA is the genetic material, but later on it was also proved that RNA is also genetic material. But what criteria or what properties of DNA and RNA makes them genetic material? That is most important. So there are four characteristics which makes DNA and RNA as them the genetic material. So the four characteristics are: first point is it should have ability to produce its own replica. That means they should have capacity to undergo replication. That means they should have capacity to form their own Xerox copy, or the similar copy should be produced by themselves. Okay. So next is it should be chemically and structurally stable. It should be chemically and structurally stable. Okay. So the molecule should be structurally stable and it should be chemically stable. That means chemically it should not be much reactive, and structurally also it should be stable so that it will not denature very easily. Okay. Then the next characteristics is it must provide scope for variation by mutation that is required for evolution. So it should have ability to undergo slow mutation. And you know that mutation is one of the reasons for evolution. So there must be scope for evolution. Okay. Then the fourth point is it should be able to express itself in terms of Mendelian character. It should be capable of expressing itself as Mendelian character. That means these molecules should be inherited from one generation to another generation. In terms of Mendelian inheritance, they should follow the Mendelian inheritance pattern and they should be recognized as Mendelian कैरेक्टर, मेंडल जिसको फैक्टर बोले थे उसके हिसाब से ये जाने जाएंगे, ठीक है। सो ऑल दीज 4 कैरेक्टरिस्टिक्स आर satisfied by both RNA and DNA, but DNA is predominantly considered as the genetic material in most of the cases. What is the reason behind it? So let us see the comparison between RNA and DNA, then only we can get the conclusion that why DNA is considered as the genetic material in most of the cases. Okay. So now we'll discuss comparison between DNA and RNA. So how they are able to fulfill all these characteristics, and which one is more suitable genetic material and why? So now DNA versus RNA. Okay. So we'll discuss the comparison between these two. The first point, that is what I said, the first point is that both the molecules should have capacity, or the molecules should have capacity to undergo replication. Okay, so the DNA and RNA both can replicate, but how they are different? See, DNA can replicate to form DNA, but RNA can also replicate, but it cannot replicate directly. ये directly replicate नहीं कर सकता. RNA can be converted into DNA, then DNA will replicate to form multiple copies, then again DNA will be forming, those multiple copies of DNA will forming the multiple copies of RNA. So here the DNA is replicating directly, direct replication, but here it is undergoing indirect replication. But however, in both the cases the criteria is fulfilled. Okay. Now next point. The next point is it should be structurally and chemically stable. So let us see how these criteria are more fulfilled by DNA. Okay. So let us see the structurally and chemically. DNA is more stable than RNA. What are the reasons? So first reason is that in case of DNA, if you remember, in DNA deoxyribose sugar is present, where in the second carbon atom, okay, in the second carbon atom OH is absent here. This is second carbon atom C2, this is C1, C3, C4, then C5. This is in case of DNA. But in case of RNA, you can see that in the 2' carbon atom, the OH group is present. And rest of the things are same in both the cases. That is here C5 is there. Okay. O, and here the base is attached. Okay. So if you compare the sugar in case of DNA, this is deoxyribose sugar. Ribose sugar. that is absence of 2' OH group. Okay, this is in case of DNA, but in case of RNA this OH is present. So presence of 2' OH group makes it more reactive. आप जानते हैं कि जितना फंक्शनल ग्रुप ज्यादा होगा उतना वो मॉलिक्यूल रिएक्टिव होगा. So OH is a functional group which is present in, this is the extra OH group which is present in ribose sugar of RNA. This is in case of RNA and it makes it reactive, and that's why it is catalytic in action. Okay, but in case of deoxyribose sugar, here OH is not there. In place of OH only hydrogen is there, because of which DNA is less reactive and it is more stable. So this is one of the reason why DNA is more stable than RNA. Okay, so presence of extra functional group in RNA in the 2' carbon atom makes it more reactive. That's why it is less stable and catalytic in action. Okay, so now what is the next reason? I think it is clear to you. Now next reason. अभी हम दूसरा रीजन देखेंगे. The next reason is that, as you know, the DNA is double stranded and RNA is single stranded. The complete DNA molecule is double stranded, but in few cases like certain DNA viruses, the DNA is single stranded, otherwise DNA is always double stranded. Okay, so the second point, second reason for stability of DNA, let us compare the stability of RNA and DNA. So DNA is double stranded. Okay, but RNA is single stranded, but sometimes it is looped. Okay, so this is DNA and this is RNA. Okay, so how this double strand is formed? How this double strand is formed? This is formed by the base pairing, and the bases are paired by hydrogen bond. Okay, here also wherever loops are there, the hydrogen bonds are there. So these are also hydrogen bond. Okay, but if you compare the number of hydrogen bond in RNA and DNA, the whole DNA molecule throughout its length is having the hydrogen bond. That means it is having more hydrogen bond. But in case of RNA, only some portion of the RNA is looped and only that portion is having the hydrogen bond. So the hydrogen bond is more in case of DNA but less in case of RNA. So the presence of hydrogen bonds to form the double stranded structure gives extra stability to DNA. So what is the reason? Presence of more hydrogen bond to form double stranded structure provides more stability, more stability to DNA molecule. Okay, so RNA is less stable. So this is in case of DNA. ये DNA में है. DNA क्यों स्टेबल है? क्योंकि इसमें डबल बॉन्ड है. तो ये डबल बॉन्ड होने से ये मोर स्टेबल कैसे हुआ? सपोज से यू आर अप्लाइंग हीट. तो हीट जब अप्लाई करेंगे तो क्या होगा? पहले DNA का जो 2 स्ट्रैंड है दैट विल गेट सेपरेट. पहले वो सेपरेट होगा, है कि नहीं? उसके बाद जो सिंगल स्ट्रैंड का जो बॉन्ड है वो ब्रेक होगा. ठीक है, ये बाद में होगा. सो डबल स्ट्रैंड होने का ये एडवांटेज है कि टू ब्रेक इट्स स्टेबिलिटी, इसका स्टेबिलिटी ब्रेक करने के लिए इसको बहुत सारे हाइड्रोजन बॉन्ड को पहले ब्रेक करना पड़ेगा. उसके बाद जाकर आपका डीएनए का जो सिंगल स्ट्रैंड है उस स्ट्रैंड में जो न्यूक्लियोटाइड है उसके बीच जो बॉन्ड है उसको ब्रेक करना पड़ेगा. बट आरएनए का मोस्ट ऑफ द पोर्शन इज सिंगल स्ट्रैंडेड, तो उसमें हाइड्रोजन बॉन्ड कम है. इसीलिए अगर कोई हीट अप्लाई होता है या फिर कंडीशन अनफेवरेबल होता है तो इसका जो कम हाइड्रोजन बॉन्ड है जल्दी वो ब्रेक हो जाएगा और उसके बाद इसमें जो न्यूक्लियोटाइड्स है उसके बीच जो बॉन्ड है वो भी धीरे धीरे ब्रेक होने लगेंगे. ओके, so this is how the stability of the DNA and RNA is measured by their double stranded nature and presence of hydrogen bond. Now next point, third reason. The third reason is that, so your third reason is, you know that children, in case of DNA, what are present? A, T, G and C are the nitrogenous base. In RNA, what are the nitrogenous base? The nitrogenous base are A, U, G and C. So here in case of RNA, in place of thymine, uracil is present. Okay, so this thymine is nothing but it is 5-methyl uracil. I have already told you. So now how this thymine, the presence of thymine provides extra stability to DNA. How? See, this is suppose a DNA molecule, okay, and this is RNA. In RNA, suppose say the sequence is A, U, G, C, U, A. This is the sequence. But in case of DNA, what is that? The A pairing with T, G pairing with C. So like this. Suppose say A, T again, then G, C, again A, T. Suppose it is present like this in case of DNA. This is in case of DNA and this is in case of RNA. Then here one thing takes place, children. The thymine can form if the thymines are present adjacent to each other. एक दूसरे के पास अगर ऐसे है तो क्या होते हैं? They forms thymine dimer. तो यहाँ हो गया और एक एक्स्ट्रा बॉन्ड, तो जिसको ब्रेक करना डिफिकल्ट होगा. सो जितना ज्यादा बॉन्ड फॉर्मेशन होगा उतना आपका क्या होगा? स्टेबिलिटी बढ़ेगा. तो आरएनए में आप कंपेयर करेंगे तो यूरासिल का ऐसे कोई डाइमर फॉर्मेशन नहीं होता, बट इसमें डाइमर फॉर्मेशन होता है. दैट्स व्हाई इट इज मोर स्टेबल. ओके, सो व्हाट इज द थर्ड रीजन? प्रेजेंस ऑफ instead of U in DNA provides extra stability by forming thymine dimer. ठीक है? So in this case also the DNA is more stable due to presence of the thymine. Besides that, one more reason is that you can see that the DNA is double stranded whereas RNA is single stranded. Okay, so here in double stranded structure the base pairs, suppose say A is paired with T by double bond, G pairs with C by triple bond, 3 hydrogen bond, and again suppose say G, C, A, T, it is like this present. And if here only A, U, C, G, G, G like this it is present. But in case of DNA, since it is double stranded, the base pair, the plane of one base pair, this is plane of one base pair, stacks over the subsequent base pair. ठीक है, इसका plane और इसका plane they are stacking together and this provides extra stability to DNA. Okay, so these are the reason why DNA is more stable but RNA is less stable. Okay, then the next characteristic was that the molecule should undergo slow mutation. It should undergo slow mutation. Children, as I have told you, जैसे मैंने आपको बताया कि DNA is less stable. DNA is more stable. Okay, so undergo slow mutation. Okay, but RNA is less stable, so undergo rapid mutation. So हमारा क्राइटेरिया क्या बोलता है? It should undergo slow mutation. And since RNA is less stable, it undergoes rapid mutation, and because of this reason, the RNA viruses evolve faster. बहुत जल्दी evolve हो जाते हैं. ठीक है? Then already we have studied that जो next point है हमारा that this is the third one. So fourth one क्या था? They should have ability to express as Mendelian character. So both RNA and DNA can express themselves as Mendelian character. So both of them can act as genetic material. But children, if you compare the characteristics of DNA and RNA, you can find that DNA is more appropriately as the genetic material because it is more stable. So to become a genetic material, it should be more stable, and that is the reason why DNA is the genetic material in most of the cases. Okay. Next, let us find out some of the differences between DNA and RNA. सो अभी कुछ डिस्कस करेंगे डिफरेंस क्या है. सिमिलैरिटीज यू नो हम ऑलरेडी बता चुके हैं. व्हाट आर द सिमिलैरिटीज बिटवीन दीज टू. सो व्हाट आर द डिफरेंसेस. जो बेसिक एंड वेरी important differences only we'll discuss. So suppose this column is DNA and this column is RNA. Okay, so DNA is double stranded, RNA is single stranded. Okay, next DNA has deoxy ribose sugar. It has ribose sugar. Contains A, T, G, C. But it contains A, U, G, C. Next, fourth point is the A pairs with T in case of DNA and A pairs with U in case of DNA. That is already known to you. So next is DNA. It is chemically and structurally more stable, but this one is less stable. Okay, so these are the some of the basic differences between RNA and DNA. And so children, with this our discussion on DNA is over, and tomorrow we'll be discussing, in our next class we'll be discussing about the RNA world, and then we'll discuss the DNA replication. ये पूरा डिटेल्स था आपका DNA एंड RNA का केमिकल स्ट्रक्चर के बारे में और उनका जेनेटिक प्रॉपर्टीज के बारे में. ठीक है, तो आज के लिए बस इतना ही. थैंक यू वेरी मच फॉर participating in the discussion. Thanks a lot.